Hello students, now we are going to discuss about the stages of drug discovery, right? So what are the different steps when the particular scientist is going to discover the particular drug which can have the therapeutic value? So what are the stages may be followed by that we are going to discuss. So stages of drug discovery, so there are 7 stages or 7 steps that the particular drug discovery company has to follow. The first one is the pre-discovery, right? Second one is the target identification. Third one is the target validation. Fourth one is the drug discovery. This is the actual one. Then fifth one, early safety test. Then sixth one, lead optimization. Then seventh one, preclinical testing. So these are the steps that particular drug has to pass, then only it can be further go for development, right? So let us discuss the first one, pre-discovery. As the name indicate, what are the work has to be done before initiating the discovery process. So first of all, understand the disease, right? Ultimately the drug which we want to discover, that is for the particular motto, particular intention, right? So what is the drug is going to be useful for, right? So that's what we need to understand, whether it is for diabetes, it is for hypertension, it is for wound healing, it is for peptic ulcer, what is the cause, right? So understand the disease, and for that particular disease, what is the requirement to overcome, to treat that disease, what is the kind of chemical moiety or chemical entity we are supposed to discover, that has to be identified first. Second one, target identification. Now once the disease is confirmed, suppose we are going to discover for diabetes, so now we have to target, like choose the molecule or select the molecule to target a drug, right? So for example, what is the cause for to overcome from the diabetes, right? It has to heal the pancreatic cells, it has to increase the beta cells, it has to able to metabolize the blood sugar level into the blood, or so many, what are the function it is supposed to be to overcome from the diabetes, that a particular drug should have that property, then only we can go for, that's what we need to target that. The third one, target validation. Whatever we have targeted, like suppose for that we need flavonoid, we need alkaloid, we need tannins, something like that. So in that, whatever we are expecting in any of the extract or any of the plant products or animal products, in that whatever the drug or molecule we have selected, we need to validate that, right? Test target and confirm its role in disease, right? Whatever the things we are expecting, like alkaloid or glycoside or tannin, that has to play the role to overcome from the diabetes, then only we are going in right path, otherwise it is going to fail, right? Then fourth one, now this is the real one, drug discovery. Find the promising molecule that could become a drug. Until and unless the particular molecule it is not proved for its activity to give therapeutic activity, we cannot say that is the drug. It has to show in the diabetic patient or into the any diabetic model, any animal model, to reduction of blood sugar and urine sugar level, then only we can say ki these are the drug that further we can go ahead. The fifth one, early safety test. Now forget about the activity, now we have to think about the safety parameter, right? First of all, toxicity is very important. It is giving activity as an anti-diabetic, but at the same time, whether it is giving any toxic effect, whether it is giving any, what is the pharmacokinetic parameter, how it is absorbing, how it is distributing, what is the metabolism parameter, how it is excreting from the body, all those parameters, all those data we need here, so that we can confirm that okay, this is now safe at least, right? Then sixth one, lead optimization. Now if it is required, we have to go for alteration, changes into the structure of lead candidate, means lead drug or lead molecule, to improve, like what is the in structure, a double bond or any functional group has to be removed, or any changes onto the structure. That's what we are studying in medicinal chemistry, like SAR, structure activity relationship. So in any modification required, so that further we can improve its therapeutic property or we can reduce its toxicity. In this way also we need to think. Then last one is the preclinical testing. Here animal model. Now these things we have done. Now directly we cannot give to the human volunteers. We need to go for the different animal models. Diabetic animals, we have to select the albino rat, Wistar rats, or their lab and animal in vitro also, it may be in vivo also, it may be animal testing to determine drug is safe for human testing or not. Toxicity study has to be done into the mice, like OECD guidelines. It is available 420, 423, 425 guidelines. There we can have the toxicity, LD50, ED50 can be determined, and those data will clearly tell the story that whether the further we can continue for the development or not, or where we are going to stop here only. That by doing all this we can be able to tell that what is the story of that particular chemical entity or that particular molecule. So these are the different stages of drug discovery when it comes into the pharmaceutical regulatory science, that you can write this answer comfortably. Thank you all.